Good morning, everyone. Welcome to today's webinar on identifying threats to forest health, an early detection and emerging species webinar for land managers. My name is Bill Jacobs. I'm the program manager for LISMA, the Long Island Invasive Species Management Area. LISMA is a voluntary association of land managers working together to prevent the spread of invasive species on Long Island and in New York City. Today's webinar focuses on the early detection and rapid response to invasive species that threaten forest health. An invasive species is one that is not native to our forests and whose introduction causes or is likely to cause economic or environmental harm or harm to human health. These invasive species may be plants, animals, or pathogens. Today's webinar will familiarize you with some of the current threats to forest health in the Northeast. You will leave with information and tools that you can use to prevent newly arrived forest pests and pathogens from becoming established, which is critical for the conservation of our ecosystems. We would like to thank our co-sponsors, New York State Department of Environmental Conservation, New York State Agriculture and Markets, the Long Island Central Pine Barrens Commission, and Cornell Cooperative Extension of Suffolk County. I would like to welcome our first speaker, Jerry Carlson, who is speaking about identifying menaces to tree health, signs and symptoms. Jerry has more than 40 years of experience in environmental health, examining insect, pathogen, wildlife, vegetation, and socioeconomic interactions in managed and natural environments. Jerry is the chief of the Forest Health and Protection section in the Bureau of Ecosystem Health and Invasive Species within DEC's Division of Land and Forests. Jerry acts as DEC's chief expert on forest protection relative to biological diversity and ecosystem sustainability. DEC's Forest Health Program provides services and related research support for private and public natural resources management, including urban and community forests, parks, forest preserves, state forests, timber supply woodlands, and private landowners throughout the state. Please welcome Jerry Carlson.
How can they tolerate changes in the climate, changes in the water, changes in the major resources that are allocated to continue these photosynthetic energy engines in a way that allows them to succeed? Can they overcome it? You know, this is all about strength. Is the strength genetic based, or is the strength a part of, you know, fertilization resources and or sun resources? You know, are they being overtopped? All of these things should hopefully enter your realm of consciousness when you're standing out there next to a tree or a forest. Say, oh wow, you know, maybe in 3 or 4 years this guy's gonna, or this girl is gonna have a little bit of trouble getting sunlight. Those kinds of things are useful ways to think about the forest and the trees all the time, so that you can sort of get better at predicting what's going to happen in the short and or the long-term future. Of course, they store starches and trees. I'm going to get into that a little bit later, but you know, obviously oxygen, the production of oxygen from the photosynthetic energy, excuse me, is what keeps most of our current biosphere going. Next. So tree decline, it's really the three major things, you know, water, the essential nutrients. I pulled carbon dioxide out of there because although it's something we would like to be able to control, we can't do too much about that, and light. So that's what good health is about from any plant's perspective. And then stress is something that limits the access to these. So these are the inciting factors. What would cause stress to this tree or this forest? And then of course, when stress occurs, you have a slowdown of photosynthesis. And when respiration exceeds photosynthesis, you have a decline in the health of the tree and or the forest. Then, of course, once you have a decline in the health, you've reduced their ability to tolerate any more major changes. You reduce their ability to fight off disease and insect agents, and those opportunistic agents are contributing factors to the death of the forest and or the tree. Next. So the other thing you have to, because we're here in New York and it is a climate change scenario, our climate factors are not easily predictable. And so every year when the trees emerge from the winter, we need to look at them and we need to separate those major weather events that caused stress and or a change in the way the health of the tree or the forest is. So, you know, we have low temperature injuries, and those are always outside the in. So that top middle picture shows a drooping, a wilting of the current year's foliage, and that's because of growing season frost. Or in the upper left there, you can see a scalding on the top from frozen winds, sun scald. And then that bottom central picture there shows dry winter wind desiccation. You can see the lower half of that little seedling or shrub is still green and the top half is red. Well, that's because it was protected by the snow level, and the part of the tree that emerged above the snow was affected by winter wind desiccation, and that could lead to a potential food source for a pest agent, or it could fully recover, but mostly those buds will die. So it's kind of put on the Sherlock cap and figure out what's going on. And down there in the lower right, that's flooding. That tree, that beautiful big green tree in the middle there, died from this, but it didn't die until the following year, and it's because it was seriously stressed by the growing season flood, its inability to move the water around at the rate it wanted to, and it was attacked by bark beetles, which killed it. Next. So this is just another example of desiccation winds. You can put your Sherlock cap on, deduce this. You can see that the lower half of the foliage is green and the middle part is red. So it could also be from salt spray, but the lower part was protected by the snowpack. The middle part was affected by desiccating winds and or salt spray, depending on, you don't usually see this kind of salt spray damage unless there's a lot of salt on the road from a corner or a hill. So, you know, you have to think about that. Also, this kind of damage is pretty ephemeral. Most of the trees recover from it. Next. So another, if your forest or your trees are near a heavy commercial agricultural area, we're starting to see a lot of these terms showing up in the forest health literature, like leaf cupping and leaf tatters. And I can't remember what that lower left one is because the screen is blocking it, but it's like shredded leaves, and these are a result of chronic herbicide injury. So best always to take pictures of stuff you see that doesn't look right, store it in your inventory, send it to someone like me or someone nearby, Cornell Cooperative Extension, and we will try to deduce what the causal factor was or go out and collect more samples. Next. So site-specific determinations, recognizing flood, wind, ice, drought, wind desiccation, storms, and soil quality. Next. So it's all about the leaves. You know, they're the primary food producers. They move the water and they make more food than they need, and that's why we have a viable maple sugar industry. They have more leaves than they need, and a rule of thumb is that they get twice as many leaves as they need. So, you know, any given tree or forest can lose up to 50% of its foliage and not suffer significant stress from that. The second rule of thumb is that early loss is worse than late. So if you ended up with more defoliation in the early part of the growing season, yeah, sure, they can recover, but they need to have the proper moisture and or light and temperature conditions to grow properly. And that's why they like to capitalize in May and June, where the temperatures are right for maximal growth and the sunlight is still there and the water availability is high. Next. So just a little bit of the leaf, you know, it's obviously not just a simple blade of green. It's more like a sandwich, and all these intraleaf organelles are critically important to the nutrition of the organisms that eat them. You have basically that spongy mesophyll is the area where the chloroplast engines are. That's where all the water's required. That's where the sunlight gets into, and the electrons are transferred inside there, going through the palisade cells. And so these are the kinds of things that are critical to the function of the leaf and the chloroplast energy producing engines. And these are the areas that are capitalized on by leaf feeding insects, especially, and pathogens too. They also want to feed on those highly nutritious areas. Next. So defoliators that feed on leaves, you know, to see in the background there, you can see that color change. We always have a lot of insect defoliators in all of our forests, most of our trees as well, but most of the time we don't see them. They have to have a population explosion before they cause that kind of color change, like you can see in the back there. We have several, this is just a few that I listed here. Lots of weird stuff showing up now, but you know, the forest tent caterpillar and the gypsy moth are often associated with large landscape level defoliations that lead to, especially if chronic defoliation occurs, that leads to mortality at the landscape level. And then we have a bunch of more or less interesting ones that show up every now and again and may cause very local mortality. Next. So one that's happened just this last year down in New York City. These are pictures taken from Brooklyn. This is the fall webworm, and you can see in the lower left there, it's quite a showy, hairy caterpillar. But most of the people don't see these because they tend to feed outside of the bright sunny day, you know, like at twilight or even early evening. But what they do is create these large webs that are easily seen. And I must have got 100 calls this last year from areas in Brooklyn where people saw them and thought we were being invaded, and which is great. And I love the fact that people bring that up, and also it's not really a problem. It just comes up every 15 years ago or so. Thanks. Next. Another one down there, you know, most of you are aware that we have the southern pine beetle, and Brian's going to talk to us more about that later. But the reason I'm bringing it up here is because the southern pine beetle doesn't always kill the tree, and the tree sometimes successfully pitches out the southern pine beetle, but the tree gets stressed. And down there in the lower right, the stress contributes to local population increases of native pitch pine beetles. And they're starting to become up behind the southern pine beetle in the material that's left over for them to feed on, and their populations get higher, and they can lead to some local mortality as well, which we're seeing. And the other picture I've got there, the oakworm and the gypsy moth, those are two defoliators, excuse me, of oak. Down there also on Long Island, and one comes out in the spring and the other one comes out in the late spring or early summer, and that double whammy can lead to a killing of the oak trees in 1 year. I've seen it down there a few times. Next. So what I'm trying to point out here is that there are things to look at, and what's more important is all you have to do is look. And if you're familiar with the trees in the forest, in the landscape you're looking at, you'll see the change, and you'll see damage to the leaves. You'll see the damage from the feeding, and you'll see new fungal infections, and you see galls, lots of galls everywhere. We get galls from all over the world in all of our trees, and they're normally not very damaging, but you know, take pictures of them, and if you don't know what they are, then let's get someone to look at them and say what the heck they are. And so it's all about change detection. You know, that upper right hand, those leaf feeding insects are causing tattering on the leaf, and you don't have to be too observant a passenger to recognize that there's something going on there. Next. So the other thing, of course, is there are animals that go out there, and you should, if not familiarize yourself with these kinds of damages, then at least take pictures of it so that someone like me or someone else can say, oh yeah, that's mice, that's rabbit, that's deer, that's a squirrel. They all, or a porcupine, they all leave a telltale tale behind of who caused the damage, if you don't already know. Next. And of course, the insects all over the place. We have defoliators, which I talked about. We have bark beetles. They all have their recognizable entry and exit wounds, like the emerald ash borer and the Asian longhorn beetle. They produce frass, which is like the sawdust that they pitch out, that they push out of their pitch tubes and or their entry and their feeding galleries under the bark and deep in the wood. All of these things are diagnostic. Take pictures, send them to us. And we can usually tell in a very short period of time what the offending agent was. Also, we have sap feeders like leaf miners and gall formers. Also, birds are in there. So recognizing the damage and symptoms, taking a picture, and getting advice on what caused the change or the stress agent is very useful. Next. So this is the Asian longhorn beetle. You get sap oozing from large wounds. On the left-hand side, those are the oviposition areas where the adult female beetle hacks into the tree, and often there's a lot of resinosis associated with that wounding. And then when the larvae are in the tree, they push out this large sawdust-like frass out of their holes as they start to bore through the wood of the attacked tree. Next. And, you know, the death of the tree doesn't have to be instantaneous. All three of these are emerald ash borer. On the left, you can see some of the woodpecker evidence, that blonding striping on the ash tree. No to see that. The tree still seems pretty green and alive, but that tree is for sure dead. If not dead now, dead within next year. And then it doesn't always kill the whole tree. It can just kill the top. As it girdles the tree, it kills it. And of course, the younger the tree is, the more rapidly it will die. And on the right-hand side, it's almost like that tree was killed by poor garden management by whipping the base of the tree, but it was in fact the emerald ash borer girdled that tree. And you can see by the size of the previous year's twigs, the shoots, how thin they are and how long they are, that tree was vigorous and healthy until it was killed by the emerald ash borer. Next. And so here's the gallery. Like you can see on the left picture, that from the very top down to the bottom where the egg was laid, the tiny little gallery, and then down at the bottom of that big J curve at the bottom where it turned into an adult. This, not much damage to the tree. The tree would overcome that. If no more emerald ash borers arrived, it would grow over that, no problem at all. But on the right-hand side, when you end up with a whole bunch of emerald ash borers, it totally girdles the tree. And then, of course, it disrupts its ability to move water and nutrients and defense compounds and everything else up and down the tree that's needed for its survival. And you wouldn't see that unless you peeled the bark away, because that occurs in 1 year. The bark is very tough and resilient to climate and or injury. So that tree would look more or less fine, except for maybe the emerald ash borer emergence holes and the fact that it had no live foliage on it. But when you peel the bark away and see all those galleries, you realize that it was girdled. Next. And woodpeckers are terrific agents of support for us. You can see on the left there, that's woodpeckers on a coniferous tree, a hemlock tree, and on the right, that's emerald ash borer. They just shred the bark looking for the insects underneath. Know how to see that, know how to identify it. You can, if you look closely at either of these slides, you can see the actual area where the insect pecked into the feeding chamber of the insect and extracted the larvae or the pupa or the young adult. Next. And it's all about location too, right? Especially in an urban environment like that sidewalk tree. Look at the diameter of that tree, and it lived for a long time with that enormous impact from root disruption. Eventually it succumbed, but obviously any landscape manager is going to worry about a tree like that for a long period of time. In the upper right, that's a landscape tree in a cemetery, highly preferred organism. The tree is, they want to retain it, but it got attacked by, I think that was oak wilt actually. But you know, not all agents are defendable given constant monitoring, but and oak wilt's one of those. Most of the ones that are not defendable given constant monitoring are new exotic invasives. That's what I wanted. But then in the center down there, I also wanted to show that another source of risk at the landscape level is monoculture. So that's mountain pine beetle damage in the mountains in western US and Canada, and you can see that all of those trees died basically in the same season. And so the insect, the mountain pine beetle, has been able to capitalize on a genetic resource, on a homogeneous genetic resource. And then in the lower right there, as people plant trees sometimes on brown fields or on, you know, especially when they're landscaping brand new sites here in New York. And this one was planted on a diesel barrel that was that was just below the surface, and it killed the tree when its roots grew into it. Next. Okay, so we need more people paying attention. We know they need to know how to report what they see. We've got forest health hotlines. You can connect with us, foresthealth@dec.newyork.government. And we're interested in anything you see that's an exotic change at the landscape level. Next. And we have a firewood regulation. Firewood's played a huge role in our outreach and education and in reducing people's movement of exotic invasive species around, not just the emerald ash borer, but several other disease agents. We're pretty sure that oak wilt is frequently moved by firewood and other pathogens. Next. Oh, can we put up the contact slide again? I think you'll see that one a few times today, but that's the next one back. There we go. Yeah. So we have a forest health hotline, but the big one that we rely on a lot is forest health, all one word, at dec.ny.gov. All of us are dec.ny.gov, and that's an email address. Okay, next, jump to, thanks, Linz. Firewood's a good habitat for insects and pathogens. Just an example. People don't often think about it, but every time you kill a tree, and leave it on the ground is food for something. And fungi and insects are very, very quick to find it and occupy it. Next. And the big thing is finding it small. If we can find the new infestation at low population densities, we can make rapid decisions. We often have the money to do it. We need less political persuasion, and we have way, way, way more success. Next. So keep looking. There's Rob with his oak wilt travel trailer. We get a lot of positive feedback from this thing. Outreach, outreach, outreach. We need to keep people, you guys who already know what you're doing, and then the people, the 99% of the people that don't, we need to keep in front of their face to say, hey, pay attention. If you see something, take a picture, send it to us. Next. And, what are we doing? We're looking at eDNA detection. Jeff's going to talk about that, I think. We're using polymerized chain reaction reproduction of DNA and sequencing for better and more rapid detection of exotic invasive species and the certainty of the whole thing. We're hoping that there will be new technologies like chemiluminescence and nanoparticle absorption. And Rob, I think, has maybe talked about the use of scent dogs for oak wilt detection and the use of scent dogs for other detection. So we're developing all these new technologies. And the last one, I think. Well, thank you very much for listening. I hope you got something from this, and I certainly did.
Thank you, Jerry.
So Bill, there's one question
Yes,
about chemioluminescence and asking if it could be explained. Individual had never heard of that method.
Jerry?
So chemiluminescence, yeah, what that basically is an altering of the wavelengths that's reflected from the surface of an organism, particularly a pathogen, which are tiny microscopic anyways, by incorporating nanoparticles that attach themselves to specific proteins. So as you probably know, protein signatures are part of the identification spectrum for most organisms, and chemiluminescence is a way to get a spectral curve that identifies that. So it's brand new technology. There are several people working on it all over the place. The University of Minnesota is looking at using it for maybe oak wilt and some other things.
Thank you, Jerry. Let's move to our next presentation, which will be on the complexities of oak decline in the Northeast with Robert Cole. Rob is a forester with DEC Forest Health and is a certified arborist. Rob earned an associate's degree in forest technology and a bachelor's degree in forestry from SUNY ESF. Rob started his career with DEC Lands and Forests in 2006. He led emerald ash borer management activities from 2009 to 2016, the Southern Pine Beetle response from 2015 to 2018, and Rob has guided the Division of Land and Forests oak wilt response since 2009. Rob co-leads the Land and Forests Project to characterize and manage white pine decline. Please welcome Rob.
All right, hello. Can everybody hear me and see my screen?
Yes, Rob.
Yes.
Excellent. All right, yep, so I'm going to be talking about oak decline in the Northeast, and I have a little bit of focus on Long Island, especially since we're doing this for the Long Island Prism. So in case anybody doesn't know what I look like, or you missed the picture in Jerry's slides, that's me in the corner there. So the first thing, if I can advance my slides, there we go. Oak decline in and of itself is a thing, but we also have several diseases that are causing the decline of the oak species across its range. But oak decline specifically refers to a complex that's brought on by climatic factors, often drought or excessive rain, plus other predisposing factors, inciting factors, and contributing factors. Jerry went over the disease spiral here already. And some of the things that are highlighted are issues that we've seen throughout the state, but also especially on Long Island, and kind of the issues that we're working with, especially on Long Island: the urban environment, soil compaction, drought in the past 4 to 6 years, bringing on all sorts of fungi and wood and bark boring insects, all the way down to the root rot, etc. So on Long Island, we have observed oak decline that we have somewhat attributed to gypsy moth and a coincidental drought that occurred 2015 and 16 especially. So you can see places we have mapped that on Long Island. And then based on some modeling done with those climatic factors that we talked about, drought and rainfall, and density of oak, etc., we kind of produced a risk map for Long Island that kind of shows us Long Island is in a pretty rough spot when it comes to its oaks. So what does this oak decline look like that we're talking about? You can see here dieback, a lot of dieback in the crown, starting with the fine branches. Then you're left with just, you know, those really, the coarse branches, the large limbs. You have the tufting of leaves, very thin crowns. It's described as a staghorn look. You'll just get the tufts out towards the end, maybe a little clump here and there, maybe some suckering off the main bowl. But this is what, if you look it up, defined as oak decline, kind of in the textbooks. There are a lot of diseases, both native and maybe not native, and some insects that contribute to this. So the rest of the talk here, we'll be talking about some specific damage-causing agents to oak. And the first one I'm going to start with is oak wilt. So oak wilt is a pathogen that moves two ways. It moves through insects, and then it moves through the roots from tree to tree. So you can see in the diagram on the left, you start out with a healthy tree. Nididulid beetles come to that tree. They're attracted to that tree naturally through wounds, and those wounds may occur from windstorms, ice storms, things like that, or they could be pruning wounds that we've created either to help the architecture of the tree or prune off a bad limb, dead stuff, whatever. These nididulids are attracted to that when they're out in the spring. And so they go to these wounds on trees and spread oak wilt spores into that. And then you see, starting over the summer, a pretty rapid kind of scorching and wilting of the leaves, starting from the top of the crown down. And then the tree will die that summer. It only takes 4 to 6 weeks to really kill a red oak. The white oak take a little bit longer. That's due to tyloses. They're able to compartmentalize the fungus better. So in the white oak group, death can take 5 to 7 years, and it'll be branch by branch. But with red oaks, and that's our big concern here, with the oaks in the red oak group, they die very rapidly, and then they transmit the disease very easily. So you have your dead red oak there, and then the following spring, when the moisture and temperature conditions are just right, spore mats form under the bark. They push up the bark, form little cracks. The nitidulids are out again in the spring. They get in those little cracks, feed on the spore pads, and then fly on to other wounded, otherwise healthy oak trees. In the meantime, when that tree has died late in the summer, throughout the fall and the winter, the fungus is able to move down through the tree into the roots, into any other oak trees that it's root grafted to. So again, white oak don't root graft as frequently as red oaks root graft. Trees in the red oak group root graft pretty readily all the time, whereas white oaks not so much. So again, concern here is the red oaks. So the oak wilt moves through the roots to close by healthy red oaks, and then the pathogen moves up through the tree, and then that summer the tree will wilt and die. So that's kind of the chart there that you see on the left. The picture that you see illustrates that in a really nice way, brings it kind of to life. So I'm just going to run through that really quick. You see the pretty defoliated tree right in the center of the picture next to the house. About 10 feet to the right of that tree was another red oak tree. Those are both red oak. And then how this all happened is this homeowner, she was getting a new roof put on her house. So she had a large limb taken off of the red oak tree that is not there anymore, that you can't see in the picture. She had that done in May. Now the spore mats have formed, they're out in May, and the nitidulids, the beetles, are also out flying and feeding and moving from tree to tree at that time period. So what we hypothesize is that there was nitidulid beetles out there with the oak wilt fungus on them. She had that tree pruned. The nitidulids could smell it. They went to the tree wound and they infected that tree. That tree started dropping leaves late July, early August. The homeowner recognized that that wasn't good. She knows that her oaks hold their leaves for a long time. But she left the tree standing there in hopes that maybe it was a fluke, it would come back. So the following spring, the tree did not come back, so she had it removed in May. Unfortunately, that gave time for the oak wilt fungus to move from that original tree down through the roots and to the adjacent red oak that you see in the picture. And they were only about 10 feet apart. So then come July again, this tree that you see here starts wilting and dropping leaves. Now the homeowner knows something is really wrong. As Jerry mentioned, there's a lot of easy indicators for homeowners. And with oak wilt, the biggest one is your oak dropping discolored and or green leaves starting in July, and it loses about 80% of its leaves over the course of July and August. So you can see how it worked out here in a real life scenario, and we ended up having to remove that tree. So as I said, we were talking about the nitidulid beetles here. So this is a little trapping survey that we did. You can see here in the center is a poster that came out of our diagnostic lab and our imaging lab. These are all the nitidulid species we've been able to trap here in New York State. We've trapped across the state. And several of these are known vectors of oak wilt. So the picture on the left, it's a little tough to see, but you can see above where the window has been cut out with the hatchet, there's a slight crack going up towards the top of the picture. And then from the bottom, there's also a slight crack coming out of there. In here, cut this out. This is where the oak wilt spore mat was. So it's very subtle, the crack on the outside of the bark. And then underneath the bark, you can see the picture in the bottom right with the dime. You can see right next to the dime, that is the oak wilt fungal mat, spore mat, pressure pad. Those are kind of different terms that are used. And that's the fungal mat that pushes the bark up and forms the crack. You can see they're pretty small They're hard to see. That crack in the bark is difficult to see. So we don't really send folks out looking for spore mats, but they are there. The spore mats grow under the bark on the surface of the wood. So they are hidden most of the time, not the easiest thing to send people out looking for. A little bit more about the nitidulids. So we did that trapping survey to find out what was out there. We also wanted to know when they were out there. So this is just a couple of species that we captured and trapped, and you can see for the most part they start coming out in May and peak somewhere either in May or the beginning of June. Some of them run a little later into July. So this lets us know when the high risk periods are, because if the insects aren't out and they're not out at the same time when the spore mats are out, then our risk of spread and vectoring is really low. So this helps us know when the high risk times are for things like wood movement and when we need to get our treatments taken care of so that we can reduce the spread. You'll see on the graph to the right of the Carpophilus species that some of them are out a little later in the year, September and October. However, because of the structure of the late wood growth on the trees, oak wilt doesn't move through the tree so easily later in the season. And at that point in time, the spore mats are no longer viable. So we don't have a big concern about that late season uptick in some of the species, but it is something to note that it is out there. So our biggest concern, like I said, is May into July with the risk of spread. So what we tell people to go out and look for, because it's the easiest thing to see, you can kind of train up anybody. It's July, it's August, our oak leaves falling on the ground. It's very uncharacteristic, it's not normal. It's a really, really telling sign that something's wrong with the tree and you might have oak wilt. So this is just the variety of leaf discoloration that you may see. The leaves come down. Generally, your discoloration is from the margins inward or the top of the leaf down, but it's not always. It's highly variable, and I just want to see you to see that from this picture. And the leaves that are falling may be falling completely green. You can see that in some of the leaves on the ground. Some may be completely brown. Some may have yellowing and green. It is pretty variable, so we just focus on the fact that they're discolored and falling. Don't worry necessarily about for the average homeowner. We're not necessarily worried about exactly what the discoloration is. I don't know how to get. So here is pictures of standing trees with the discoloration that we're talking about and the leaf drop that we're talking about. Remember I said about 80% leaf drop. It stands out. These trees are both surrounded by other oaks that are retaining full crowns with green leaves. So it's very easy to see. Again, if you're working with a homeowner or you're managing a property, you go around and look for this type of discoloration in July and August. It's maybe one of the more simpler ways to go about looking for it. So where do we have oak wilt in New York? Well, I've mentioned Long Island a little bit. We have it out in the Finger Lakes area, Canandaigua, South Bristol, Middlesex. We call that the Canandaigua Lake area. It's on both sides of the lake there. And then we have it in Glenville, which is in Schenectady County, just outside of Albany. And we have 4 locations on Long Island and 1 in Brooklyn. In general, oak wilt first described in the 1940s in the Midwest. You can see the map in the upper left. It's still moving out there. There was a big expansion of oak wilt in the 80s, kind of coincided with large expansion of suburbia out in the Midwest, and we think that's probably due to construction damage on trees, wounding of trees, bringing in those netidulid beetles, helping spread the oak wilt. So you can see it's since moved. It's in the western half of Pennsylvania, a little bit more. There's some counties there that you see that aren't colored in Pennsylvania that are along the New York border and surrounded by red areas in Pennsylvania. And those are probably also positive for oak wilt. It just hasn't been completely tracked in that area. So we are worried about spread out of Pennsylvania for sure, especially in Western New York. In Long Island, the story there is it's a little complex. Going back to the title of the presentation, the story there is complex. It was originally found, reported by an arborist to the Department of Ag and Markets. They worked with Cornell University to get a couple tests done. Then DEC got involved. We were not able to culture oak wilt in the traditional way at any of these sites on Long Island. We were able to confirm it using PCR and DNA sequencing. So we found it in this neighborhood. This is Central Islip. We visited about 100 properties here. We went door to door. We inspected all oaks in this area. This is about a half-mile radius of the original trees. We found all the red points you see there are positive trees. So our treatment there and at all the other sites, the site in Southold out on the North Fork was one individual roadside tree. The point you see to the west of the North Fork is in Wildwood State Park. That was a single tree. And then West Islip is down on the South Shore, and that was an individual tree as well. So all of this happened over the course of one field season, so we expected to find a lot more trees. So our treatment on Long Island was to remove the infected trees. That way we couldn't have that spore mat production, we couldn't have the netidulid vector. But assuming we would find more because we found it spread across all of Suffolk County, we didn't want to go into a lot of tree cutting because we didn't think we had the full picture. So we only removed infected trees, and since then we have found no new infected trees on Long Island, trees infected with oak wilt. So we're not sure why we didn't see a lot of that root graft spread. We expected there would be root grafting and that we would find it in other locations, but we did not. That's not to say it's not out there. We are constantly looking, constantly doing aerial surveys. It's a real, it's kind of a mystery to us, and we continue to investigate, but it's not what we expected to happen. The tree in Brooklyn, that was also an individual tree in a cemetery. Again, origin kind of unknown, and no new trees found in that area. So what do we do when we find oak wilt infected trees? We write up individual site management plans because we have to deal with not only the spore mat and the netidulid vector, but we have to deal with the root grafting and root graft transmission. And those things are going to be kind of determined site by site. So you can see here's a map. This is the house out in Canandaigua that we looked at earlier. Tree 2 is the one that was standing in that picture, and tree 1 is the one that was missing from that picture. So we go out on site and we measure the diameters of the infected trees and then all the nearby trees. And we use this table up in the upper left that predicts the distance that oak wilt could travel through root grafts in a given growing season. So we measure, and so what you do is you take the combined diameters of, say, your infected tree, tree 2, and tree 7. You combine them, and then you look at the table, and it will tell you that distance that oak wilt could travel, and that if they were root grafted, it may get it. So what we do is, the first thing we do is we put in our trenches to disconnect all those root grafts. And we include healthy trees inside of our trenches, our root graft disruption, to make sure that we are capturing all of the oak wilt that could be in the roots, in the ground, within our trench. Because between tree 2 and 7, you don't know exactly where the roots are, and you don't know how far the oak wilt has moved through the ground. So you don't want to put your trench in between 2 and 7 because you might miss it and tree 7 may already have oak wilt. So we trench outside. You can see those yellow lines. Those are where we trenched in this instance. And then the trees inside those trenches, inside, closer to the infected tree, are removed. We do the trenching first. We do the trenching and then remove the non-infected trees, and then we remove the infected tree last. We don't want to lose the pressure created by the tree. When we cut the tree down, it lets everything go out into the roots, and you can actually facilitate the quicker spread into the roots if you remove that tree first. Sometimes we install a physical barrier into the trench. In this instance, the homeowner had tree 8, you see there, big, beautiful, 26 to 30 inch red oak shading the south side of the house. We didn't think it was highly likely that tree 8 was root grafted to tree 1 or 2, but just in case, we wanted to make sure we put that trench in and then put the barrier in so that tree 8's roots could not grow into possibly infected roots of tree 1 or tree 2. The trench is only good for about 5 years before roots can start growing back through it. So if you put this barrier in, you guarantee that the roots won't go and reconnect to those old infected roots. We have had new infections start by roots growing into old infected stumps 5 years later. We had that instance at one of our Glenville sites. We cleaned it up in 2009, and then in the fall of 2013, we had a new infection there because of root graft transmission. You see we go down pretty far. We get buckets that are 5 feet so that we can make sure we're breaking all root grafts. Our management of oak wilt does include a bit of a regulatory aspect. When we have just one infection center in an area, we put a half-mile buffer around it, and then we use roads and property lines to create what we call a quarantine district. And you can see that in the bottom right. This is our Brooklyn site, and all the squiggly lines there, those are the roads going through the cemetery. And then the solid green line is just following streets around the perimeter and then out into the actual city streets a little bit there. So with the quarantine district, what the quarantine district does is it regulates the transportation of oak wood and firewood out of the area. It doesn't regulate any practices such as pruning or harvesting timber, but it does regulate the movement of any of that wood out of the quarantine district. So for instance, the cemetery, if they take down an oak tree and they can't get it chipped up on site because it's too large, they have to call the New York City DEC office, forester, and get a permit from him to be able to move the wood out of that quarantine district. Once we get a couple of these, what we call neighborhood quarantines in a town, then we go to the town level. So you can see that in the town of Glenville, up on the larger New York State map. And then once we get a couple of towns within a county, we go to a county-wide quarantine district. And you can see Suffolk County does have an oak wilt quarantine district established for it. So from March through July, we do not permit the movement of oak wood out of Suffolk County or any of these quarantine districts. And then for the remainder of the year, if you would like to move oak wood out, you should be calling your local forester and getting a transportation permit. We also include firewood in this because most folks can't identify small firewood-sized pieces of firewood to species, so we include all firewood. So again, in those spring, early summer months, we don't allow the movement of firewood out of the quarantine district. and then for the rest of the year, it does need a permit. These activities that we have on this green card in the upper right-hand corner, again, the risk is high when the nitidulids are out and the spore mats are out. And that's, April roughly into June, and the nitidulids are out a little bit later. So we consider that the high-risk period. So if you don't have to prune oak trees during that time, please do not. If you have to, say there's a storm and you need to make a corrective pruning cut to a broken branch or something, paint it. And now normally we don't recommend painting wounds anymore, and this is kind of the exception for oak wilt, and you can use a regular can of spray paint right off the shelf from your local hardware store just to cover the wound so there's no access for the spores to get into the tree. That should about cover oak wilt. Oh, right, we have the dogs. So Jerry mentioned the detector dogs. So these are a couple of the dogs that we're working with through the New York, New Jersey Trail Conference and the Lower Hudson Prism. And here we were out for some training. You can see on the left there, the dog is alerting to finding oak wilt in that tree. We actually had a small container of oak wilt culture, the fungus culture, hidden up directly above where the dog is sitting, up in the tree branches. And he was able to circle the tree a little bit and alert right where our oak wilt was in that tree. Then we took him out to a couple field sites, and you can see the stacked logs here and the logs sitting on the band mill there. These logs are in a forest that is one of our oak wilt sites where there is oak wilt, and the dog was interested but did not alert to any of these logs. But it was really interesting. You see the band mill. The dog got up on that log, climbed up on top of the band mill, spent probably 5 minutes sniffing around there trying to figure out if there was oak wilt there. So we see a lot of potential here. If we let some logs move and then we find out later they may have had oak wilt, we could send a dog out to a sawmill or other locations to go track down any of the fungus that may have spread to new locations. All right, so that covers oak wilt. So I'm going to talk about some other damage-causing agents that we have that might present signs and symptoms that look similar to oak wilt. The first one is Anthracnose of oak. Now, I mentioned the discoloration from oak wilt is later, mid to late summer. We're talking mid-July into August. But there's a lot of other things that cause discoloration and leaf issues on oak. So Anthracnose is one of those. You can see the picture on the left. You have these tattered looking leaves. Well, actually both pictures. Tattered leaves kind of look water soaked. They're blotchy, just distorted looking. This symptom you'll see earlier in the season. This is often May into June, and it's caused by wet springs, and it can be really bad in springs when we have a lot of rain because it's windborne, but also spreads in water droplets, raindrops. So once you get it going in the spring, if the spring continues to be wet into the summer, it can really prolong it. But because it's an early, it causes damage to the leaves early on, the trees normally can reflush, and this isn't a super big deal. If you get a really serious infection, you can get infection on the twig and then get twig mortality, which can bring in secondary insects. So then we kind of get talking back into the compounding factors of the disease spiral. So you can bring in, if it coincides with things like gypsy moth, you can get armillaria in there, drought. It can be a factor. Generally it's not though. It just doesn't look good in the spring. It dries out in the summer. The leaves reflush and everything is fine. So we don't really do any treatment for this. You just watch it. Keep keep, for individual trees, yard trees, street trees, trees you're managing in a park, it's about individual tree health, and trees can get anthracnose several years in a row, and it doesn't cause huge problems unless you have those other factors going on. Another pathogen that we have that discolors leaves is Bahia leaf spot. And this discoloration coincides a little bit closer with oak wilt than, say, something like anthracnose. But the discoloration is different. Now, this is usually, July, August, maybe even worse in August, discoloration. So you see it starts with spots, small spots with concentric rings that go out, and as the tissue dies in the center, and then you have that brown ring, and then that dies, and you end up, it all coalesced and can turn into one large brown spot, which from a distance, you could say, maybe from the ground looking up in a tree, that could look like oak wilt, but when you bring it down closer, you'll notice all these spots that have kind of come together, these black-brown spots. This is also something that can be moved by wind and rain. So again, it's those wet seasons can be really bad for this sort of thing, but this is later in the season. And it's usually newly established trees or trees that are otherwise stressed that really get hit by this hard and can succumb and maybe have some mortality. Otherwise, a healthy, mature tree, this is just a late season kind of blight on the leaves that is not going to have a long-term health impact. Two-line chestnut bore, we haven't really talked about any insect damage yet. Two-line chestnut bore, you can see here, it was a pest of chestnut. Now that most of the chestnut's gone, it's a major pest. It can be a major pest of oak. You can see where the name comes from. It's got two lines down the elytra. You may remind you of emerald ash borer, close relative, both agrylus. What it does is its larvae feed underneath the bark, essentially girdling the tree, and you'll end up with scorched leaves at the top because they're not getting any water. Now, this could look a lot like oak wilt, except the difference generally is that with oak wilt, you lose 80% of the leaves. With two-line chestnut bore, the leaves often stay on the tree the remainder of the season, so you don't get that leaf drop like you do with oak wilt. And that's one of the big diagnostics, especially in the upper Midwest, that they use. Rather than doing lab tests, they say, oh, all the leaves are still hanging on the tree. It's two-line chestnut bore versus all the leaves have fallen off. It's oak wilt. So two-line chestnut bore, again, that's a native. It's around. Its population really jumps up after gypsy moth and or drought. And so I mentioned that Long Island has experienced both of those things. And we've done some survey for two-line chestnut bore down there, we haven't been too successful in trapping a lot, but we suspect a lot is out there, and it's kind of the final nail in the coffin often for some of these stressed oaks. It's something to watch out for. Again, this is individual tree health. Make sure trees remain vigorous, and if it's in a situation where trees can be watered during drought, water the trees during drought. This is one that we often get people calling confused with over oak wilt. This Cagney Axia, if I'm saying that right, I prefer the name hypoxylon because it's a lot easier to say. But these cankers form underneath the bark as well, and they start out dark color, almost black, and as they coalesce and they grow and get larger, they push the bark off and they turn this gray or silverish color. But people often call us and send us pictures of this and ask if it's oak wilt. This is not oak wilt. So remember, oak wilt will enter healthy trees and kill healthy trees. This canker is the tree is really stressed. It's on its way out. These trees are going to be very unhealthy. They may have little to no crown left in them when you see this. So that's another kind of feature you can look at. A healthy, vigorous tree, you're probably not going to find this on. It's at the later stages, it becomes very obvious that the tree is dying. Bacterial leaf scorch, another leaf discoloration. This one can be confused for oak wilt quite easily. One of the main differences that we tell people to look out for is you can see the picture on the left between the brown scorched area and the green. There's the ring or halo of yellow, a yellow band through there. That's one of the big diagnostic features and differences between bacterial leaf scorch and oak wilt. Oak wilt generally does not have that yellow ring, although I showed you some pictures where there was some yellowing. So with bacterial leaf scorch, the symptoms, you often don't see them until the end of August or September. These symptoms show up usually a little bit before regular leaf change. So while the discoloration looks very similar to oak wilt, you see that oak wilt discoloration earlier in the season than you see the bacterial leaf scorch. That's not to say that the symptoms can't overlap, but if you're out looking at trees in beginning of September and they look like this, they have a water-soaked appearance, you may have bacterial leaf scorch. And we can test for both bacterial leaf scorch is present in New York State. It's New York City, Long Island, up the Hudson Valley. So we know we have it. So there's, when we take oak wilt samples later in the season, we often test for both so that we can make a determination in case it comes back negative for oak wilt. We could still tell somebody they have bacterial leaf scorch, or we can say you have oak wilt, not bacterial leaf scorch. So we look for both, but those symptoms they appear later, generally in September. So then we end up with something like this, and this is back to the complexities of oak decline and where we get a little stressed out sometimes. So this sample, you look at it and you go, okay, some of these areas look water soaked, maybe there's the yellow halo, but it is scorched from the outside inward. Which is it? Is this oak wilt? Is this bacterial leaf scorch? And in this particular sample, it's actually both. So you can have a mix of pathogens or damage-causing agents going on. In the tree, you can have more than one thing happening. So we don't, again, like I said, when we've been talking about this leaf discoloration, we use it sometimes to hone in on what might be going on, but in an instance like this, better test for both, better see what's really going on there. So we will sample just about anything if we're not sure, and then when there's, our symptoms getting covered up by another thing, is it oak wilt, is it, what's going on here? So it does make it very complex, and the treatments are different, and we don't want to go cutting trees down that have bacterial leaf scorch because they often recover. Again, this is a late season defoliation, so the tree's pretty much done with all the photosynthesis it was looking to do for the year, so not a big deal. So we don't want to make a mistake and cut down trees that we're going to recover thinking that they have oak wilt. So our diagnostics are very important. Gypsy moth, I brought that up already a little bit in the decline. So you can see the gypsy moth egg mass there, about 1 inch wide, maybe 2.5 inches long. Cottony, fuzzy, this one here, the eggs have already emerged out of. You can see the holes. It's a bit tattered. It's kind of old. It'll start out a little bit lighter, cleaner looking and smoother. And there's the European gypsy moth there in that picture with its characteristic set of blue dots on the back and then the red dots on the back. We've had gypsy moth here, since the mid-1800s. It's cyclical. Again, if that cycle coincides with a drought or other issues, gypsy moth can cause mortality. Generally, oak can sustain a few years of moderate defoliation, but if you throw in a drought or you throw in one of these other pests, it could do the tree in. One of our concerns, so we have the European gypsy moth, is the Asian gypsy moth. You can see in that picture in the bottom right, they are very similar looking. However, the Asian gypsy moth has a wingspan of 2.5 to 3 inches, whereas the wingspan of the European gypsy moth is under 2 inches. The European gypsy moth female does not fly. She lays her eggs wherever she can walk to. The Asian gypsy moth, however, flies the female, so could spread quicker, and it has a larger host range. So we're always on the lookout for the Asian gypsy moth, but we are not aware of having it currently. You can see heavy defoliation can occur. This is a picture taken at the end of June in Orange County. This was several years ago. All those trees 100% defoliated. And out, I can't remember exactly where that picture in the bottom left is from, somewhere south of Rochester, but that's a spruce tree. Again, several years ago, went to a site visit. Homeowner called in about spruce defoliation, covered in gypsy moths. Just that one tree. There were other trees around it that didn't have any gypsy moth on it, but gypsy moth can be a pest of trees other than oak, and it could be spruce. And to wrap things up, please use local firewood. New York State has firewood regulations still in place, as Jerry mentioned. No untreated firewood into New York State, and firewood within New York State, if it is not treated firewood, I mean heat treated, it cannot move more than 50 miles, and you need to fill out a certificate that you can find on the DEC website. And as Jerry mentioned, pathogens can move in firewood. This is a picture of a piece of firewood at our original Glenville oak wilt site that has the remnants of a spore mat on it. And that is it from me.
Thank you, Rob. We have some questions. First question is, what is the exact name of oak wilt, the scientific name?
It used to be Ceratocystis fagacearum, but it's now Bretzellia fagacearum.
Okay, and the spelling for that, I assume people can find online.
Yeah, the spelling Bretzellia is Bretziella.
Wonderful, thank you. Can you talk more about the Brooklyn infestation of oak wilt? Was that confirmed?
Yes. So Brooklyn was confirmed. That that all happened the same year as the stuff in Suffolk County. We confirmed that. Again, not sure where it came from, but that cemetery there in Brooklyn is, you know, you can see all the ports and the ships and everything there. There's a lot of stuff moving. A lot of people, believe it or not, do move firewood around kind of locally there in that area. We're not sure where it would have come from, but we did confirm it there. It was in one tree, and that tree actually was the tree Jerry mentioned it. He showed it in his presentation. It was pretty isolated. There were no other oaks around it, so the cemetery had that one tree removed. And we continue to monitor there every year and do our netidulid trapping. But there has been no new detections in Brooklyn.
Okay. And we've posted in the chat the spelling, if anyone's interested, for oak wilt. The trenching, we had a couple of questions about the trenching. How deep are the trenches for oak wilt?
We go down 5 feet if we can, or if there's bedrock or something we can't get through, we go as far as we can, but up to 5 feet. We want to make sure we get all the roots if we can.
And on the table, Rob, the distances in, were they in feet or inches from the trees?
Okay, yeah. So it's feet. So those diameters in on the left column, those are in inches, And so it's the combined deviation. So if you have two 12-inch trees, it's 24 inches, and then it'll give you the distance the oak wilt could move in feet. So if the trees are within, you know, I don't have the table back up in front of me, but say it says 40 feet. So if those two trees are 40 feet or less apart, that means the oak wilt could move between those trees. And then that's that we would choose to remove that tree.
Thank you. These, I'm not sure what these, but these only apply to mature older oak trees, or can seedlings be affected by these? I assume maybe oak wilt and some of the others.
Well, oak wilt can impact any age oak, and in any of these diseases or insects that I talked about really could. The big thing is maybe when asking about age is stress level and overall tree health. So a lot of these, yeah, you'll see, you know, a lot of these are kind of secondary stressors that compound each other. So, you know, older trees, unhealthy trees that can't fight, they don't have, you know, the energy to respond to these things. Yeah, they'll go first, but it could be young trees, newly planted trees that aren't being watered enough, or, you know, just the shock from planting could also be at risk.
Okay, and what fabric is used to line the trenches?
Oh, you know, I wish I could remember I'm not sure the exact name of it. It's basically a heavy landscaping fabric, and it's water permeable, so the roots will grow up to it and not grow around it. There were some studies where they used like sheets of rubber or other impermeable barriers, and the trees would actually grow up to it, and still they could, you know, they could tell that there was water on the other side of the barrier, so they actually grow along the barrier and around it and back to where they could find a water source. So it's an impermeable barrier, or it's a water permeable barrier that we use, but I don't know the exact name of it off the top of my head.
Okay. Any other questions for the chat? Well, thank you very much, Rob. We appreciate you being here. Our next speakers, Tom Allgaier and Michael Giambalvo, will present on spotted lanternfly, early detection, reporting, and agency response activities. Tom is the invasive species coordinator for the New York State Department of Agriculture and Markets. Tom has more than 20 years of experience working for state and federal agencies on invasive species and plant protection issues. Tom has worked in various landscape construction, garden center, and greenhouse positions. He holds an associate's degree in ornamental horticulture from Farmingdale State University, a bachelor's of science degree from Empire State College, where he studied environmental biology, and a master of science degree from the University of North Carolina, Wilmington, where he studied environmental science. Michael is an assistant horticultural inspector for the New York State Department of Ag and Markets. Michael has spent the last year inspecting trees on Long Island and in New York City for signs of the invasive Asian longhorned beetle. Michael was recently transferred to the Albany office as the Spotted Lanternfly Program Education and Outreach Coordinator. Michael has worked in various gardens, farms, labs, and seed banks, and holds a Bachelor of Arts degree from Warren Wilson College, where he studied environmental science, sustainable agriculture, and biology. Michael is currently enrolled in a master's program at Stony Brook University, where he is studying geospatial science. Please welcome Tom and Michael.
Hello, everyone. This is Tom Allgaier. Michael, are you on? Okay, I wasn't sure if Michael was going to be able to join us. It sounds like he was not. So I'll be flying solo. As you can see from the slide, it's New York State. I work for New York State AGM. It's the new preferred official policy. It's not Asian gypsy moth. I just wanted to let you know. It's agriculture and markets. I know with this crowd, you see AGM and automatically you think, reflect back to Jerry's talking, you think of Asian gypsy moth, but it's our new acronym. For those of you that don't know me, this is me. I'll spare you the horror of actually seeing live video. So I think most of us are familiar with spotted lanternfly. I'll just briefly touch on the biology. It's a plant hopper native to China and Southeast Asia. It was first discovered in Pennsylvania in Berks County in 2014 and has spread to multiple other counties in Pennsylvania, Maryland, parts of New Jersey as well. They use their mouth parts to feed from sap. They use plant pressure. They don't really suck the sap out. They just basically the plant sap pressure pushes the sap through their body, and they have a lot of pass through. I'll talk more about that later when I talk about the honeydew issue. They feed on about 70 different plants that we know of. They have a preferred host of Ailanthus, which is also known as tree of heaven, another invasive species. The biggest concern are grapes, hops, apples, maples, walnuts, also some ornamentals and forest plants such as sumac, oaks, and they have been found to feed on a few pines. And here you can see the fourth instar nymphs here on the right, which looks like they're feeding on what looks like a very young tree of heaven. You can see the notches in the leaves in the bottom right at the base of the leaf. But you can tell that that's not sumac, that it's tree of heaven by that leaf notch. So the impacts, as I mentioned earlier, the feeding stresses plants, makes them more vulnerable to other diseases and other insects that are opportunistic of stressed plants. They excrete a honeydew, which is basically their excrement mixed with the sap from the plants. This in itself causes issues. That sweet excretion attracts stinging insects. The way it was first found in Berks County, that honeydew gives off like a fermented smell. As the sugars start to break down, it actually started to ferment. So the property owner started noticing this fermented, almost like an alcohol smell, and a higher abundance of yellow jackets and other stinging insects that were attracted to that. And then one thing led to another, and they found this insect. You can see here in the top feeding on apples. They don't feed on the fruit. They feed on the stems, just as they do in grapes. You can see them all on the adults feeding all on the grapevine, but not on the fruit. On the right, you can see an adult with its wings partially spread. You can see all the blackness on the leaves there. That is the black sooty mold that grows on the honeydew. That will cover everything below it, including houses, cars, pools, recreation equipment, children's toys, your deck. Makes it very unpleasant to be outside. If anybody remembers back to the 80s, to the gypsy moth outbreaks, and you go outside on a bright sunny day like it is here in up in the Albany area, it would sound like it was raining. That would be the excrement falling down from the gypsy moths. Well, spotted lanternfly is very much the same. It's such a high population, such a high density population, the excrement will fall down and it'll feel like rain on a bright sunny day. So it has, it hinders outdoor activities. It's a quality of life issue as well as an agricultural and forest pest. It's all around just a bad actor. Here's what the lateral view of an adult looks like. Not quite that large. They're about 3/4 of an inch to an inch long, but they're pretty consistent in coloring. They don't really look like many other things. They're a very large tree hopper, plant hopper, sorry. You can see a little bit of the reddish coloring of their hind wings coming through their translucent forewings. So it gives them a bit of a pinkish hue, but when they spread their wings, it's that bright photo that everyone's used to seeing with the moth-like image. So the life cycle right now, the stage that we're at is the eggs are starting to hatch. The nymphs are beginning to emerge. We've had a cold spring. So really, late April was really pushed into May. Even down on Long Island, you had some cold weather. So the life cycle is going to be delayed a little bit more likely. But it's very similar to what it is in Pennsylvania. So when the nymphs first emerge, they're black and white, like this center photo here on the bottom of the page. They'll molt 3 times, so they'll go through 3 instars, each time getting a little bit larger, but they'll remain black and white. The 4th time that they molt, they'll have red wing pads, and it's very distinct. At that point, they're a little bit bigger. They're about pea size. They're a little bit easier to spot in the environment, especially with the red wing pads. Early on, when they first hatch, they're very much like the size of a small deer tick. They're often confused for ticks at that very first instar. They're very hard to spot in the environment. They disperse upon hatching, so they're very hard to spot in the environment in the early nymph stages, but as they develop, they're a little bit easier to spot. As the summer begins, they'll mature into, they'll shed their exoskeleton one more time. They'll emerge as adults in the top center there, and then they'll continue to feed. And then they'll mate towards the end of the summer, early fall, and they'll lay eggs. The eggs are not always covered. As you can see, that there's 2 photos of eggs there. On the right, there's several rows of eggs that have not been covered. Even the eggs on the left that have that putty kind of glossy covering, similar to gypsy moth, but it's more of a smooth, glossy surface, which will dry and crack and lose its glossy covering. But that waxy covering is a protectant from predation. It blocks pesticides and things like that. But even there, you can see that the eggs at the top are not completely covered. So you can't always look for the covered eggs, and they're very difficult to see. You can see how that blends in with the bark, especially as it starts to age. The egg masses are very difficult to spot. They have a tendency to lay eggs underneath things on the bottom side of things where they're a little bit more protected and harder to spot. They also have an affinity for rusty metal for laying eggs. They'll lay eggs on any flat surface, anything that's not moving, pallets, stone, grandma, if she sits in the backyard too long. But what we really worry about is that rusty metal. So equipment, campers, trailers, construction equipment, construction supplies like I-beams and things like that, they're easily and readily moved around. They don't transport themselves very far. They prefer to hop and walk. They can fly. They're not really strong flyers. They're moderately, they can fly, but they're not going to fly for miles and miles. The way that they get dispersed quickest is by human-aided movement, whether in vehicles or attached to material as an egg. One of the things that we're doing is we're surveying. I think everyone's familiar with that. What we're doing for survey is we're using sticky bands like this bug barrier here to the right, where one of the characteristics of the insect is that in the nymph stage especially, they'll crawl up and down a tree. They'll crawl up, they'll fall off, they'll crawl back up. So they're up and down the trunk multiple times a day. And in that process, they'll crawl underneath that plastic barrier, get stuck on the sticky material, and be collected there. I mentioned a little bit about visual survey, especially for the eggs and the adults. I'll get a little bit more into the other 2. So we have partnered with Lower Hudson Prism and the New York, New Jersey Trail Conference. They have 2 dogs that are trained for spotted lanternfly, Dia, and I cannot remember the other dog's name. But we've had them out for survey work and also for some regulatory work at roadside checkpoints. The New York State DEC has been conducting some environmental DNA or eDNA sampling, where they can sample, take DNA samples off trees to see if spotted lanternfly had been present in the past 3 days. It's weather dependent based on on moisture and things like that. It's a new technology being applied. It's more often used in aquatic environments, but it's starting to be used in terrestrial environments as well. So we're partnering with them as well. The surveys are conducted in 1 kilometer grids, as here's an example from New York City, Queens area. So each pink grid there is 1 kilometer square. We inspect up to 10 host locations. So a host location would be preferred tree of heaven, but we'll also look at wild grape. We'll set up traps, and the most important thing is reports from the public. We've done quite a bit of outreach. Michael Giambalvo is now on board, just specifically for spotted lanternfly outreach, to get the word out there and get the information out there. It's a very striking insect. As with most invasive species, we're more likely to hear it coming in from reports from the public than our survey crews, just because the volume of land mass to cover and the small number of survey staff. What they may see in the field, as I mentioned before, uncovered eggs is the bottom right here. She's only laid a few strips there. The completely covered egg mass on the upper left on that Ailanthus trunk. You can tell it's Ailanthus. It has almost like that cantaloupe skin quality to the bark of the trees when they're younger. And then there's the adult female getting ready to lay some more eggs. We also look for the nymphs, although the first few instars are much more difficult to detect in the environment. They're more striking when they get those red wing pads, as I mentioned earlier. So primarily, these are the things we're looking for visually when we survey. The dogs can detect egg masses as well as adults. Unintended transport, as I mentioned before. Here you can see on the front pillar of this pickup truck, there's a spotted lanternfly just waiting to get it, jump in that window and go for a free ride. The right side here, you can see the rear end of a tractor trailer around the brake lights. There's several egg masses underneath on both of those rusty metal surfaces. That's the sort of thing that we fear the most. There are permits. We have an external quarantine in place in New York State. Department of Agriculture put that in place shortly after Pennsylvania had their expanding issues. We will accept the Pennsylvania permit, or any of the states that have it, will have their own permitting systems, but the kind of the gold standard is the Pennsylvania permit. It's a 2-hour free training. Anybody moving material in and out of the quarantine will need any material that could possibly host spotted lanternfly will need a permit to either stop in a quarantine or transport material from the quarantine to here being New York. The 2-hour training for the permit gets into a lot more detail of that. Again, this is just an update. It's not an overall, extensive SLF training. I mentioned the detector dogs. Here you can see Dia working with the handler from Lower Hudson Prism, alerting at the grill of that vehicle. And it's very hard to see, so we circled it, but there is a dead spotted lanternfly in the grill of that truck. Very, very easily missed by the human eye. The detector dogs have a much better rate of detecting spotted lanternfly, especially in some of these harder locations to visually inspect. As I mentioned before, up underneath on the underside of things. If the back end of that truck was parked, we can inspect with inspection mirrors that reflect up, but the dogs do a much better job. Another technology that's being employed, we've partnered with DEC in the past, as well as New York State Department of Transportation and New York State Police at the roadside checkpoints. Some of these are at rest areas, some of these are at DOT way stations. We'll use unstaffed aerial vehicles to inspect loads that are unsafe or just in the effort to speed things along. So here you can see the drone over the top of the truck inspecting the stone. There's also a log truck here. We wouldn't want to be climbing around that because the logs could shift and it could be a dangerous situation. What we're looking for again is like this bottom right photo here was a spotted lanternfly that one of our inspectors, Jamie Lynn Woodside, found. And then this center photo looks very much like an egg mass, but it's actually just a grease spot. But that's the sort of thing that we want to spot and investigate further. I'll go through really quickly. Initially, when we started doing the roadside checkpoints in 2018, we found very little compliance. We've done quite a bit of outreach since then, and the compliance rates have greatly increased. We're hoping to see that trend continue. We will be conducting more checkpoints this year, although a reduced number due to COVID restrictions. Survey as well is been curtailed as of right now due to safety reasons and other duties, because some other more pressing matters occurred. And so spotted lanternfly field survey will continue throughout the year very shortly. As far as more information, more specifics about spotted lanternfly, the best resource is the spotted lanternfly at agriculture.ny.gov website. There's a plethora of information there. There's many physical handouts, trifolds on different, whether it's the permitting or whether it's just general biology or questions and answers. We have a fact sheet. There's lots of information available from that website. But the key thing is currently there are no known established populations within New York, and we really need the public's help in reporting. And that's all I have. If there are any questions.
Thank you, Tom. Our next speaker is Jessica Cancelleri, who will present on white pine decline and disease. Jessica is a research scientist with DEC Forest Health. Jessica manages the Forest Health Diagnostic Lab, which provides diagnostic services and research support to public and private land managers. Jessica holds a BS degree in wildlife science from SUNY ESF and a master's degree in entomology from the University of Nebraska, Lincoln. Jessica has been with DEC since 2010. Please welcome Jessica.
Everybody, can you hear me?
Yes.
Great. All right, well, it's great to be here today, and thank you for taking the time to listen in. I'm going to talk about white pine decline and disease, which is a huge, complex landscape level issue. Back in approximately 2009, widespread health issues started emerging on the landscape for white pine. It seemed to start in New England and then move its way down into New York. And in the DEC, we first started noticing some issues around 2012, 2013. I remember, driving to sites for other things and noticing some discoloration of pines on the road and thinking it was perhaps salt damage or winter injury. But then it became obvious really quickly that there was something else going on. So now it's been about 10 years that a lot of people have been studying this phenomenon, and we've learned a lot. And it's now been shown that multiple fungal pathogens, an insect pest, and a changing climate are responsible. So lots of factors that we're dealing with here. So what is white pine decline exactly? Well, throughout the eastern US and Canada, white pines have experienced unprecedented damage in recent years due to previously innocuous native pests and pathogens. So that's a key factor here, is that we're dealing with serious health issues that are a result of pests and pathogens that have never been known to cause damage before, or at least not cause significant damage. And so why are they causing damage now? What has changed? And these combined factors are severely impacting white pine growth, productivity, and economic value. So although the symptoms vary depending on what complex of pathogens or insects are present, they generally include the things that I've listed on the screen. So yellowing, browning needles, premature defoliation that occurs in the spring rather than the fall, really thin canopies, like you'll see in the picture on the right. That's really characteristic of what we see all over the landscape. stunted shoots and needles, sometimes resinosis, and of course branch dieback. And for the first several years of this phenomenon, we didn't see a lot of death. This is a slow decline, but I'd say starting in about 2016, 2017, we started seeing pretty significant mortality at some of our sites. So to really consider what's happening with white pine, you really need to think about land use history. It plays a huge role in what's happening right now. So you have to consider what's happened on our landscape over the last 100 years. And you know, white pine has been influenced by things such as intensive logging, right? So, you know, around the late 1800s, throughout the the latter half of the 1800s, well into the 1900s, most of New York State was logged, and white pine was the primary lumber species that was harvested. And so all of our best, mature, straightest, most beautiful trees were taken. And what was left was, you know, the scrabbly, bent, you know, unhealthy trees that nobody wanted. So for one, our genetics have changed dramatically. You know, the same genetics are not flowing through the system as once were. When widespread planting occurred later in the 1900s, during like the 1930s, the CCC movement and putting people back to work, you know, a lot of these areas were replanted with white pine, but those seeds came from the Midwest or the South or from Europe and did not represent, you know, the genetic diversity that was already present prior to the logging. So you also have excessive field abandonment, you know, in which as our society shifted from an agricultural society to a more industrial society, a lot of people gave up farming, and those fields were colonized by white pine, which is an early successional species. It takes advantage of disturbed habitats and colonizes readily on them. There was also, you know, as you know, a lot of fire suppression over the last century, and you know, white pine regeneration benefits from periodic fire. So regeneration has been hindered significantly. And then of course you have the introduction of non-native species, such as white pine blister rust on the landscape, which when that came on the landscape was probably the most destructive disease that we've ever seen in white pine. And then of course a changing climate. So with all of this in mind, then you add on to it, you know, just this new suite of unexplained factors. Again, why are these pathogens and insects that have once been considered harmless now actually causing damage to our white pine? So what agents are responsible? Well, there's a lot of them. So like I've mentioned a couple times already, the native pests and pathogens. The largest one contributing to white pine decline right now is white pine needle damage, which is a complex of 4 different fungal pathogens. And I'm going to talk about that a little later on. Caliciopsis canker, another native pathogen, and white pine bast scale. These are all natives. Environmental stressors, drought is a big one, and increased precipitation. So these are all likely a result of climate change. New York sees more rainfall than it ever has in the spring months when these diseases are producing spores and when white pine is flushing its new needles. And we also see increased drought. And the thing is over the course of a year, you know, the total rainfall or the total amount of drought necessarily hasn't changed that much, but it comes in these really discrete periods. So we get excessive rainfall for 2 weeks, and then we get a drought, and then we get excessive rainfall again, and then we get a drought. So it's just happening in discrete periods, which are stressing the trees even more. And as a result of the land use history that I talked about on the last slide, there's a lot of white pine currently that's just growing where it's not supposed to. It's on bad sites, either because it was planted there during the CCC era and it wasn't really considered, you know, maybe we didn't know quite as much as we do now, or it just came up on this, you know, crappy, compacted old farmland. And so another huge one is stand density. And you're going to hear me talk about density a lot in this presentation because it's really one of the largest factors that you can control that mitigates impacts of diseases in white pine. We have a lot of high stand densities in New York, and that's largely a result of, you know, in some places white pine is easily sold and the lumber market is good for it. In other parts of the state, they can't sell it. So if they can't sell it, you know, they can't harvest it. So we'll talk about Caliciopsis first. This has probably been around for millennia, but it was really first noticed in 1880. And it was noticed causing damage early on, but very small localized damage, like little clumps of trees here and there. Nothing to really take note of. You can recognize Caliciopsis by, it's a canker. So it causes cankers on the main stem of the tree. And they're kind of reddish brown in color, and they're real soft. So if you push your thumb into them, they'll depress into the tree. One of the most obvious signs are these black fruiting bodies called parathecia that are present year round, and they look like eyelashes. You can see a picture in the lower right-hand corner of that. And up above is a macro image that we took in the lab of the fruiting bodies. And even more obvious is the severe resinosis that you can see in the picture in the upper left. And that's mild compared to some cases we see. So Caliciopsis is associated with overstocked stands. There's been a lot of research done that shows that its impacts are much more severe in dense stands versus thin stands. Suppressed and pole-sized trees are more susceptible, but in landscapes, trees in dense groves or screens are most at risk. Trees can have many cankers, and the cankers degrade both the vigor of the tree and the lumber quality. Regenerating saplings in the understory are particularly infected, which can result in mortality. Mature trees are a little bit more resilient and can withstand an infection better. But we've been to places in New York where we've seen a high percentage of the regeneration wiped out by Caliciopsis. Most recently, it's been found that Caliciopsis is attacking stressed and weakened trees in association with feeding by another insect called the white pine bass scale, which is on the next slide. Oh, no, I guess it's not. We'll talk about that in a little bit. So this slide is basically showing that infection by Caliciopsis causes a reduction in the functional sapwood at the site of the canker damage. So the canker kills the xylem tissue near the cambial area, and therefore reduces the functional sapwood cells. So that reduces the vertical transport of water and nutrients and reduces the tree's growth and reduces its overall vigor. So on this figure here, the black points indicate bolts that were sampled from trees in thin stands, and gray points indicate bolts that were sampled from trees in unthinned stands. So as average canker length and associated damages increase within a bolt, the ratio of sapwood to heartwood decreases. So I just mentioned the eastern white pine bass scale, and this is a really interesting story too, because the white pine bass scale is a scale insect that is native to the Northeast. It's been known for a long time to be from Eastern Canada and New England, not so much in New York, maybe further up north. But in 2007, it was first detected in the Southern Appalachians and since has been found throughout the Southeast, which is weird because, we're used to pathogens or insects moving northward as a result of climate change, but we rarely see that movement south, especially from a northern dwelling insect. And this is an insect that has never warranted any major attention from entomologists before. The name of it refers to the tissues that are affected by the scales piercing and sucking mouth parts. Bass cells are a part of the phloem and contain fluids just beneath the outer bark. So while other species in this genus have been known to be serious pests of pine, white pine bass scale feeding results in almost no damage to infested white pines. So, well, what's the big deal then, right? Well, it's because this association with Caliciopsis has been documented. It's been shown that the pathogen takes advantage of these feeding sites of the bass scale. It allows them an entryway into the tree's vascular system. That was much harder for them to achieve before. And so, these scales are typically found in association with the cankers. So they're just allowing the establishment to happen like much more rapidly and at much more of a larger scale than before. So researchers believe that, you know, a sort of facultative relationship has developed, and they've actually labeled it like a pest pathogen complex. The bat scale, though, is really difficult to ID in the field. It's absolutely tiny. You definitely need a hand lens. The second instar, which is the cyst stage, is the most conspicuous and the easiest to identify. It's shiny black in color. It can be found near branch whorls, within cracks, and especially underneath lichens. And the insect is found on all size classes of eastern white pine in association with Caliciopsis. So there's also, you know, just some native wood rotting pathogens that are sort of, you know, ubiquitous on the landscape that I won't talk about for long, but I just wanted to mention, you know, armillaria is a root decay fungus, and it particularly hits sapling and pole-sized trees. Mature trees can usually withstand it pretty well, but that's not always the case, depending on what else is happening or how stressed out the tree is. We have seen large numbers of mature trees succumb to armillaria. Phaeolus Schweitz, I don't know how to pronounce this one, Schweinitzi, is a velvet top fungus. You know, wood in advanced decay appears dark brown. It readily breaks into like these cubical sections. It's a cubicle rot. The brown rot fungi like Phaeolus result in dramatic reductions in the bending strength of the wood. So typically the fungus doesn't end up killing the tree. It's, you know, mechanical damage like wind breaks and, you know, wind damage and stuff like that, that will eventually make the tree succumb. And red ring rot, this one's a little more destructive even than the others. We talk with foresters about it all the time, and it seems to be the bane of the existence of many foresters because it's so hard to detect. And by the time you've detected it, not that there's much you can do anyway, but you just don't see it until it's had quite an effect. For mature white pines, red ring rot results in significant loss of timber and managed forest. And the pathogen invades through branch stubs, but the decay is confined to the heartwood. So symptoms are, like I said, are usually too nuanced to really notice, but these punk knots might be visible on the trunk, which you can see in the photo there. You'll notice like a swelling and bulging of the trunk, and you may see, you know, the fungus growing out of it. So annual conchs and like perennial conchs are not reliable indicators though of decay, and they tend to be uncommon. So the one that you see in the picture there, like I said, you'd be more likely to see the swelling than you would to be able to, you know, actually see the conch. White pine blister rust. So, you know, this is a rust fungus that's, like I said, probably been the most destructive disease of five needle pines until now. Its origin is Asia, but it came to North America via Europe in the late 1800s, early 1900s. And as most of you probably know, it has two alternate hosts, right? Ribes, which are currants and gooseberries, and white pine. So it needs both of those hosts in proximity to complete its life cycle. So back starting around maybe 1920 and lasting for many decades, there was a large-scale eradication effort of ribes in an effort to control white pine blister rust, which was, you know, really economically damaging to the lumber industry. And it seemed to be relatively successful for a while, but then starting in the 1990s, we saw an increase in reports of white pine blister rust. And there's been a lot of studies done recently that are looking at the resistance of supposed resistant cultivars. So, you know, during this period, like post-eradication, you know, there was a lot of pressure from, you know, agricultural investments or, you know, interest to get more species of ribes back on the landscape again. And so there was a huge production of resistant and supposedly immune cultivars. And now we're kind of starting to see that those cultivars aren't so resistant anymore. And the incidence of white pine blister rust on the landscape is increasing. So, you know, when I first came on the job, I guess I never thought this was still going to be an issue. I thought this was something in the past that we talked about and, like, you know, story to tell. But But now a lot of us are starting to pay a lot more attention to it, and foresters are looking for it in the field and reporting it to us. It may end up being a problem again. And symptoms of white pine blister rust, like the most obvious is probably the actual orange rust fungus and the general orange color of the cankers. The fungus starts on the needles of the tree, so you'll often, this symptom largely goes unnoticed, but you'll see like a yellow spot maybe on the needle, and then it moves into the stem where it causes these cankers, and then it causes a branch flagging and branch dieback. You'll notice, like in the picture at the bottom there, kind of a swelling of the branch. It's sort of like a barrel-shaped, spindle-shaped swelling. You'll see excessive resinosis as well. And it is possible for the rust to move into the main trunk of the tree and cause mortality. So telling apart Caliciopsis and white pine blister rust can be a little tricky in the absence of the orange rust fungus. The main things that we look for is with white pine blister rust, the resin typically comes from one location, and it's typically located at whorls. And I say typically because there are exceptions to this. Whereas with Caliciopsis, it's just sort of all over the place. It's like a bunch of small cankers throughout the bowl, and cankers tend to be between whorls. And of course, like I said before, with Caliciopsis, you have the stalked parathecia, the eyelashes. With white pine blister rust, you have the orange-yellow blisters. Another major difference is that the rust fungus is most visible in the spring, but with Caliciopsis, you can see those parathecia, the eyelash fruiting bodies, year-round. So, white pine needle damage. So white pine needle damage, like I said before, it consists of 4 different fungal pathogens, which we'll look at in a little closer detail on the next slide. Even though the symptoms vary depending on what particular species are present in any given stand, most of the pathogens result in premature needle shedding. It's important to note, though, that these diseases rarely kill trees by themselves. And yet the annual cycle of infection and the loss of older foliage, it creates a chronic stress that eventually weakens the trees, and eventually they can't recover. So the photos in the bottom kind of show the progression of the disease from May till June. So this is perfect timing right now on the landscape to start looking for this. This is the time of year that you can really see white pine needle damage. And then at some point it kind of gets hidden and you don't notice it anymore. So right around this time of year, we'll start seeing massive discoloration of 2nd and 3rd year needles. Current year needles are not really affected. So the needles turn color, then starting in, depending on the weather, maybe mid-June, you start seeing significant needle drop. And if you have a lot of white pines near you, it's very obvious. I mean, we've seen sites, where cars are covered with them, or, we went to a community one time that was covered in white pine, and their whole tennis court was covered in like 2 inches of white pine needles. And they had said that they had actually cleaned them up the week before, and they had been replaced within a week. So it's very obvious. If you're in a forested setting, you'll mostly see them on top of the understory, like ferns. And once needle drop is over, the resulting effect is like the picture on the right. On the right is a tree that hasn't been hit as hard by needle damage, and on the left is a tree that has. So you've lost all the older needles, and now you just have the current year needles on the tips, which give it sort of like this candlestick appearance. So let's look at the needle diseases a little bit closer. So Lecanisticta acicola, that's a brown spot needle blight. Many of you have probably heard of it. This is that and Septoriotes strobi, which are the 2 on the right. Those are the most common fungal pathogens that make up white pine needle disease. They're ubiquitous. They're everywhere in the landscape. We find them in every stand. Bifusella linearis and Lophophacidium duxii are also common, but a little less common than the others. And again, especially Bifusella and Lophophacidium, these are again 2 pathogens that we've known about forever, but they've just never been known to cause problems. Lecanisticta, the brown spot needle blight, has been known to cause problems in the past, but records like from the last 100 years indicate literally like tiny pockets of mortality of, 7, 8 trees here and there. So this is a really new thing. And, the jury is still sort of out on Septoriotes strobi. I think, they're starting to come to the understanding that it's probably a native fungus, but when it was first found, it was actually first found in New York in 2015, and there was no known records of it in North America, and its closest relative was on Japanese black pine in Japan, not the Japanese black pine that we have here. But I think since that, they've learned that it most likely is a native that we just did not know about, or perhaps was misclassified. So this work by Broders et al., again, just kind of shows the timing of the symptoms that I outlined earlier. So right now, you will see some needle necrosis right before needle drop occurs. So as defoliation is occurring, you'll notice some needles are fully yellow or already dead. Some you might just see, half the needle is brown and half the needle is green. But these symptoms are obvious now. And then chlorosis is more obvious in June, and defoliation only happens in June. So it's interesting to see how trees are compensating for white pine needle damage. I think right away, the symptoms that we were seeing, it sort of was apparent that these trees were just spiraling towards death, rapidly, but we're finding that's not the case so much. A lot of research has shown that they're increasing their rate of photosynthesis to compensate for the reduced foliage that they have as a result of the spring defoliation. And they're also prioritizing carbon storage over growth. So they're holding on to that carbon so that they can use it in future years. But unfortunately, that's limiting their growth. So, maybe that's not so much of a big deal for white pines on the landscape, but it sure is a big deal for foresters trying to manage and sell their timber. So I thought this study was really, really interesting because it looked at the impacts of seasonal litter fall dynamics. So what's happening is in June and July, they found that the defoliation of the white pine accounted for 47% of the total annual litter fall. So those of you who are familiar with white pine, you know that they naturally drop their needles in October. And so, they're not accustomed to dropping them in the spring. And as Jerry, I think, pointed out earlier in his presentation, when defoliation occurs earlier in the season, it has much more severe impacts. So it's resulting in a really significant nitrogen loss because the nitrogen content of the needles in the spring are much higher than the nitrogen content of the needles in the fall. So they lose that nitrogen and then they're unable to uptake it during the rest of the growing season. And this has been shown to severely affect the growth. And in this particular study, they showed that severely infected trees reduce their basal area increment 25 to 73% compared to pre-outbreak years. And that's huge. So I want to talk a little bit about the work that we've been doing in DEC. And that's me up there and Rob Cole on the left. You heard from him just prior. Him and I are co-leads on this project. So starting in, I think, 2017, we established about 30 monitoring plots statewide with the goal of assessing stand characteristics associated with white pine needle damage. So we wanted to know, what types of stand characteristics made needle damage more severe or not. So, we took a bunch of tree health measurements and collected, things like basal area. And on the bottom right, you'll see some of the results from that. And the red points are, trees per acre and basal area per acre for all of the sites that we took measurements from. So the important thing here to note is the take-home message is that all of our stands are very dense. You can see the managed C line at the bottom. You want to be, beneath that for mitigation of white pine needle diseases. And most of our stands are well above that. So we're going to talk about this a little bit more, but low density management is critical to mitigate the disease impacts. So This slide just shows a sample submission to Cornell and the accompanying results. I like to just highlight this because it shows the ubiquitousness of these needle diseases. This was, one site in New York that we sampled, but this is literally what it looked like for every site that we sampled. We collected needles and sent them to Cornell for analysis, and they got back to us with the particular species that were found on them. The ones that I've circled here are ones that are associated with white pine needle damage. And there's many others that are found in addition to the white pine needle damage. Here's some results also from our work that shows another relationship between stand density and white pine needle disease impacts. So the first figure on the left, we have crown density and we have white pine needle disease rating. So as crown transparency and white pine needle disease rating. So increased crown transparency is associated with increased white pine needle damage, right? And if you look at the figure on the right, the basal area of the stand, as that increases, as stand density increases, crown transparency also increases. And here's some summary results from the tree health measurements we took. So LCR is live crown ratio, CD is crown density, CT is crown transparency, and WPND is white pine needle damage. So that was measured on a 0 to 3 scale. So everything else is in percentages, but then the needle damage severity is 0 to 3. So this was only the first 2 years of long-term monitoring. We don't expect to see that much change from year to year, but we did, unfortunately. Live crown ratio increased though almost 5%, and that's a little bit confusing because you would think if these trees are undergoing any kind of stress that they would not put on any new growth. But we explained that in the data because a lot of these high levels of live crown ratio increase were actually associated with a lot of mortality of trees around the ones that we measured. So trees were dying in the stand, and it was allowing more light into the stand that allowed these trees to put on some growth. But overall, it was a decline. Crown density decreased 9% in 1 year on average. Crown transparency increased almost 10% in 1 year. And for white pine needle disease rating to increase almost 1 unit is pretty significant. We also did an analysis based on our work. We have some partners with a group called the White Pine Alliance, and Bill Livingston is part of that, who is a forester from the University of Maine. And he created this D-score measurement, which is used to detect the start of declining growth. And it measures BAI, which is basal area increment, right? How much growth does a tree put on each year? And so these are just some examples of the samples that we took from various sites in New York. And you can see decline just with your naked eye over to the right. To the left is, the previous history of the tree. And as you move right, you see that the rings are much closer together. But to quantify that a little bit more, we can look at this table real quick. So this is a list of all of our sites. And on the right is the D max, which means looking at the entire history of the white pine core of the tree, the D max identifies the period of time in which decline was at its highest rate in the entire history of the tree. And it's really interesting because for all the sites that we did, we found that Dmax to be post 2010, exactly when these pathogens really started becoming obvious and its associated stress on white pine in our landscape. So, like I said, Rob and I, we've been part of this White Pine Alliance for quite some time, and the goals of the White Pine Alliance are to really share with partners the goals of low density management and the role of low density management in mitigating disease impacts from white pine needle damage and all the other associated factors. So a big part of this is just knowing where it's worth it to focus on white pine and where it's not. And so soil is a big part of that, right? Like white pine likes deep well-drained sandy soil. You know, you want to avoid excessively drained soils, and especially soils with rooting restrictions. So in a lot of these abandoned agricultural fields, you know, we see these really dense layers of soil, you know, a few feet down that were the result of years and years of plowing, you know, and working the land. And what happens is it almost turns into this thick, you know, dense, almost like clay layer, and water sits on top of that. And then the white pine roots hit the water, and they sit in water, and they don't like wet feet, and they're severely weakened and stressed as a result of that. So, you know, I know this can apply not only to a forestry perspective, but also to an urban perspective. Everywhere you drive, you know, you see rows of white pine, or, you know, we see it also with things like Norway spruce. You know, you see those really dense sort of hedgerows, or, you know, wind guards that people plant with highly dense trees next to each other. And if people can start thinking about this a little bit, even in an urban landscape, you know, if you just thin out a little bit and you give the trees a little bit more room and a little more resources, they'll do a little bit better. You know, we can't take away these diseases, but we can help the trees respond better to them. And unfortunately, you know, this is such a landscape level problem. There are no other control methods other than low density management. So, you know, we see this with homeowners as well. You know, sometimes we'll visit sites, for example, in the Catskills, you know, where people have a little bit of land, and maybe they live in the city and they come up, you know, to enjoy their forested property in the Catskills. And it's, you know, 90% white pine, and it's all dying. And, you know, it looks like the picture that I showed you at the beginning of the slide. And, you know, a lot of people like that think that, you know, more trees are better. And so that's a big part of what we do, is just trying to convince even just the average homeowner that more trees aren't always better. You have to take out some of those trees, which is not always easy. But, you know, as we move forward with white pine management, we're just going to have to keep these questions in mind. Chances are that, you know, the distribution of white pine is going to be decreasing in the future. It's going to be, you know, these diseases are going to restrict the distribution, and we're going to have it on the best sites and where it's supposed to be. And I think eventually on the poor sites, and, you know, in areas where we've inappropriately planted it, it's going to succumb to the diseases. So I don't think this means the end for white pine. It just means a shift in how we've known it currently. So this is a slide that just shows our reporting information for the lab. This is where you can go to ask questions about white pine. White pine is our most highly reported thing at the lab. From about 2015, it became one of our most highly reported problems. So a lot of people do see it on the landscape, especially between the months of May and June. So you can email us or call us with questions, or if you're a land manager that has a sizable amount of white pine that is having problems, you know, we'd like to know about it, and we can potentially give you some management recommendations and work with you. We're also doing some research in the future. There's a couple things that white pine is struggling with in New York right now. One is stands that are too dense, and the other is lack of regeneration, right? So we need to think about the next generation of white pine and make sure that we are promoting the growth of the new generation of white pine. You know, we have all these old mature stands that have no regeneration in them. So we're going to have some research goals, and if any of you, you know, might have small stands that are, you know, that could be potential research sites, we might be interested in talking to you. And I think that's all I have. So it's a little bit early, and I'd be happy to take any questions.
Thank you, Jessica. We have a couple of questions here. First one, when thinning stands of white pine, has there been any active restoration of other native species on the site that you are aware of to fill in the gaps?
Well, in the situations that I'm aware of, in terms of, you know, the objectives of the White Pine Alliance, they are managing purely for white pine. So for example, in some stands, they will actually use herbicide on hardwoods to suppress hardwoods because, you know, stands that are good for hardwood growth are bad for white pine because eventually the hardwoods outcompete the white pine. So in this case, they are looking solely at white pine. There may be, you know, some management of invasive species and stuff like that in the understory. And this is actually something that, you know, Rob and I have talked about because we don't know that we want to necessarily do that in New York, or if foresters would be on board. Do we want to manage 100% for white pine? Probably not. You know, we have a lot of beautiful stands that are a mix of white pine and hardwoods, and we'd like to find a way, you know, we'd like to sort of tailor the management strategies to meet our own objectives in New York. So we're not really sure, you know, what that would look like yet on our end, but like I said, historically, it's the focus has just been on white pine.
Okay. Jessica, can you go into more detail on the relationship between crown transparency and stand density? Some people
Yes,
were wondering, weren't completely
of course.
clear on it.
Yeah, that's good. So white pine needle damage, you know, when a stand is dense, we have shown that denser stands result in higher levels of white pine needle damage. You know, the fungal pathogens do better. And that's for a bunch of different reasons. One, the trees are overcrowded. They're competing for resources. They don't have everything of what they need. Perhaps they're not on an ideal site, you know, and also they're closer together. So these fungal pathogens, you know, they're dispersed through rain and wind, and even internally through stem flow inside of the tree. So when they're close together, you know, they move around much better. So as a result of white pine needle damage, the trees become weakened, and they lose their needles in the spring, as we said, up to, you know, 40%, 47% of needle loss happens in the spring. And so that is actually what causes the crown transparency that we're talking about. Once these trees have been repeatedly defoliated, you know, you look up and they have a small fraction of the needles that they once had. And so that lets, you know, more light into the canopy. It also reduces their photosynthetic capacity significantly. So crown transparency, you know, is a bad thing. And going back to those graphs I showed earlier, you know, an increase in white pine needle damage is associated with increase in crown transparency. It's not just because of the defoliation of the needles though, however, it's also just a general weakening of the tree as a result of years of disease, you know, and defoliation.
Okay. Well, thank you very much, Jessica.
My pleasure. Thank you.